I'm Barbara Rosario for ISSNAF Stories, and I'm with Professor Pietro De Camilli, the recipient of this year's ISSNAF Lifetime Achievement Award. Pietro De Camilli is professor of cell biology and neuroscience at Yale School of Medicine and Howard Hughes Medical Institute investigator. Many congratulations, Professor Camilli, for the award.
Thank you.
And let's jump right into talking about your research. Could you give us an overview?
Yes, I work at the interface between fundamental cell biology and neuroscience. Cell biology is a discipline that studies the basic mechanism of function of all cells. Neuroscience is a discipline that studies the function of the nervous system. And neuroscience is a big umbrella that comprises many different subdisciplines. Some neuroscientists studies how the collective actions of neurons generate cognition, response to the extracellular, response to the external world. And the cellular neuroscientists like me study the mechanism through which individual cells support this function of the nervous system, and also how the fundamental properties of all cells have been adapted through evolution to support this highly sophisticated function. This is a very exciting field because by studying the molecular and cellular properties of the nervous system, we are also close to understanding the molecular mechanism of diseases. And in particular, throughout my career, I've been interested in the dynamic and properties of the intracellular membranes. Cells are highly compartmentalized by membranes. This membrane defines subcellular organelles. These membranes are made of lipids. And I've been particularly interested in understanding how these membranes support the function of the nervous system in synaptic transmission. Synaptic transmission is a mechanism through which one neuron sends signal to another neuron. This signal is represented by chemical substances called neurotransmitters. Neurotransmitters are contained into small sacs or vesicles in the presynaptic compartment and are released in the space between two neurons by a process of fusion of their membrane with the outer membrane of the cells. And we have been very interested in understanding how these membranes are made, how they traffic within cells, how they acquire the lipids. These are lipid-based membranes, as I said before. Our modification of the lipid control the fate of these organelles. And I would say that my major specific contribution to this field is really to bring the role of lipids and their modification in the dynamic of these membranes. And obviously this work had brought us has not only helped us to contribute to understand how the nervous system works, but also provide us some general insight into mechanism of importance for all cells. This is why I've always worked at the interface between the discipline of cell biology and neuroscience. And in fact, I spent most of my career in a department of cell biology. I was even chair of that department for some years. And more recently, I moved to neuroscience. I've also been chair of the department of neuroscience. So I really been at the interface of the two fields.
And you and your group recently published a study that could have implications for new therapeutic targets for Parkinson's disease, a study that I read, it's on trash cans of cells. So what can you tell us about this?
Yeah, actually, I would say by serendipity, because this is not something we expected, two of the proteins that have been at the core of our work on synaptic transmission and on the properties of this neurotransmitter containing vesicles, it turned out to be Parkinson genes. And with this, I mean that extensive genetic studies over the last couple of decades have led to the identification by other group, by human geneticists, of genes whose mutations are responsible for familial form of Parkinson's disease. So these are genes whose mutations are responsible for generating Parkinson. And the next goal of this field is to elucidate the function of each of these genes, to understand how their mutation results in disease. And for example, an interesting question is whether the function of some of these genes is interrelated. And so since two of the genes that have been shown by genetic studies to be implicated in Parkinson are genes which are very familiar to us because we have identified and studied them. So we have become very interested in applying our basic research to the study of Parkinson. And in the study that you mentioned, we have found that an organelle which in the cell is responsible for eliminated degraded substances or substances which are old, which are damaging from the cell, they need to be eliminated, what can be called the trash can of cells, are organelles. So we have found that one of the proteins that we are studying is actually implicated in the function of this organelle in maintaining their integrity. And so we think that when there is a mutation in one of these proteins, the integrity of this organelle, which is called the lysosome, what has been defined as a trash can of the cell, the membrane of the organelle lose their integrity, their content can spill out in the cell, damage the cell. And so now we are interested in ways through which this dysfunction can be prevented. I should say that there is increasing evidence that dysfunction of this organelle plays really a critical role in Parkinson's disease. And so our studies of these processes are central to what in current direction in Parkinson's disease research.
Is there anything else in the general fields of cell biology or neuroscience that you're most excited about?
Well, I think what excites me is in general is how we are close to understanding the molecular mechanism of diseases. There has been a dramatic convergence of different fields in the last several years, in particular after the sequencing of the human genome. And the advances which have been made bring us really close to understand in detail the molecular mechanism of each disease. And so this understanding mechanism is the premise to identify therapeutic target. And so it's really an exciting moment in biology because there is really a convergence between basic mechanism of medicine. I should mention that when I started my profession many, many years ago, if scientists that study fundamental mechanism of cell function were very far from people studying diseases because we didn't have any way to understand to understand in molecular mechanism diseases. Human genetics has provided so much information about gene implicated in diseases that often in modern time medicine precede basic science in understanding the function of different genes. So this convergence is really spectacular, and there couldn't be any better moment to be in this field.
Would you be able to quantify how far are you from out of developing a therapy for Parkinson's disease, for example, or other diseases based on your research?
Let's say we are very far from identified mechanism that can be therapeutic strategies that could be applied generally to all form of Parkinson's. However, because now we start to understand the function of specific gene, we are relatively close to identify ways through which Parkinson's disease, due to specific mutation, could be treated. So, for example, there is a disease in a protein called LAR2, which is a protein kinase, and the mutation activates this kinase. And so the idea is that by developing inhibitor of this kinase, we might treat the form of Parkinson due to this condition. And what's very interesting is that, for example, our proteins, is not this one, is a protein which in the case of our protein, is loss of function rather than gain of function that create Parkinson. However, it acts at a site very similar to where the protein LAR2 function. Also, LAR2 is implicated in lysosome function, what are defined as the trash can of cells. And so if you understand better the interconnection of these different proteins, perhaps we can identify additional therapeutic target that may help to treat at least subset of Parkinson's disease.
Let's change subject. You were born near Citiglio, near Varese, studied in Milan and Pavia, and then moved to the United States for a postdoc at Yale, where you went on to become a professor. Did I get
Correct.
everything right? Correct.
Correct.
Yeah, and then I also read that you then returned for a few years to Milan and then back to Yale. So I'm curious about this short return to Italy, which I think is quite unusual.
Yeah. So yes, I was born close to Lago Maggiore, and that's really the place which is in my heart. So I spent 80 every year when I was young. Every year when I was young, I spent 8 months in Milano during the school year, and then for 4 months I would be in the countryside, and that's really what shaped me in a many different ways. And those 4 months, what made me enthusiastic about learning about nature. And I think what I do now is really an extension of the curiosity for nature I developed in those early days. Um, anyway, so I was educated in Milano. I was lucky to go to a very good high school called Liceo Manzoni, where I made many friends, which still remain friends as of today. And then I went to medical school. I really wanted to be a scientist, but at the time, we didn't have a PhD, it was many years ago, and I felt like I would receive a better training in science by going to medical school. In particular, what I like about the idea of go to medical school is I will learn only about one organism, but at all level of analysis from molecules to mind. And I never regretted this choice. So during medical school, I started attending laboratories in the Department of Pharmacology and a Center of Cytopharmacology supported by the National Research Institute. And I had really fantastic teachers there. For example, Jacopo Dolesi, that was my direct mentor, Francesco Clementi, Bruno Ceccarelli. It was a fantastic environment to work with. And when I graduated and I finished my first research project, I decided to come to the United States for a postdoc. I landed at Yale in the Department of Pharmacology, where I worked in the laboratory of Paul Gringer, that then went on to win the Nobel Prize in year 2000, I think. Shortly after arriving there, another Nobel Prize, George Palade, that had been the mentor of my mentors. The people I work with in Milano had been tutored by them, recruited me to the Department of Cell Biology as a faculty, again at Yale. So both Gringer and Palade were at Yale. I had some reluctancy to do that, to accept this offer, because I thought that really I want to come back to Italy. But George Palades convinced me. So I stayed on for a short while, but then I say, no, the pull of my country is too much. I felt like I owe to my country to go back, and I did. And but then after a few years, after these 5 years, I realized that after having been exposed to the science I could do at Yale, it was very difficult for me to readjust to the situation in Italy.
And where were you in Italy? What were you doing?
Milan, again in Milano. Yeah.
At the university, professor in Milan.
Yeah. I was an associate professor at the University of Milan, yeah, in the Department of Pharmacology.
Mm-hmm.
So then also I had to say my wife is of Italian ancestry, but lived most of her life in America. So she was in favor of coming back to the United States, and I did. And I went back to Yale, and I've been there ever since. First, until 2015, in the Department of Cell Biology. And after 2015, I moved to the Department of Neuroscience. There was no specific reason. They wanted me to become chair of the Department of Neuroscience, so I made this transition. But I retained a full appointment in the Department of Cell Biology, so I'm really equally citizen of both departments. I would say that one of the most my rewarding experience has been in 2005 to to be the co-founder of a program in cellular neuroscience, neurodegeneration, and repair. And the goal of this program, through which we could recruit several wonderful assistant professors, was to really help to push the boundaries between cell biology and neuroscience. And this has been a program that's been very successful and really fit with what I told you before, an effort to bring together the two disciplines. On the one hand, to understand how fundamental properties of all cells apply to the nervous system, and on the other hand, to take advantage of the specialized features of the nervous system to understand cell biological mechanism. And in between all this, there is a possibility to understand mechanism of diseases.
Let,
In particular neurodegenerative diseases, which are really the next frontier.
let's talk about ISSNAF, of which you were one of the founders. What does this award mean to you?
It means a lot because there is in the one of, in the gospel there is a sentence of Jesus Christ that say nobody propheta domo suas, I mean nobody is a prophet in his own village, and so it is special honor to be recognized by my fellow citizens. So this meant really something. It was something really special for me.
ISSNA works to connect scientific research between Italy and North America. So how do you see yourself contributing to that bridge?
I always kept a lot of contact with my Italian colleagues, in particular the colleagues in my field. I do believe that science is international, and so it is natural that scientists move around. Uh, if you are in science, you want to be in the places where the major advances are being made, where you have a very important community. And so it you cannot do that without being some flexibility, having some flexibility to move. However, not everybody can move, and so it's important to keep this connection. And until recently, the United States one of the best place to come. I think, unfortunately, unfortunate for us, fortunate for other people, maybe the trend can change. But it's natural and important that some Italians are in these places and they keep contact with people in Italy to function as catalyst for the exchange of information. So it's really critical. And I kept very strong contact with the Italian scientific community. And one way in which we at ISSNAF contribute to Italian science is by being part of scientific committees, a review board, evaluation panels, and um, the, so it's always important when you want to evaluate a structure, a particular scientific institution, to have people that understand the institution, but also to have direct connection or direct conflict of interest. And so we, the Live Abroad, have been often used by the Italian scientific community in these endeavors. And I participated in many of these endeavors, Teleton, Armenese Foundation, the IIT of Genova, more recently the Technopole, which is a very prominent institution. And this has been a wonderful way to maintain in contact, and I hope to have contributed a bit to Italian science.
What do you like the most about living in New Haven?
Well, I I work in New Haven, but I live about half an hour away from New Haven, in Guilford, which is a small town on the shore of the Long Island Sound. Uh, it may seem strange because New Haven is an ugly city, but Guilford is a beautiful little town, and I feel fortunate to live in a beautiful state, in a beautiful place. I lived for most of my life, with the exception of what the time I spent next to Lago Maggiore in Milano. And at the first was strange to live in a community so different from a big city, but I ended up enjoying it very much. It's a very simple life. I don't feel the need at weekend to go places because I live in the places where people come during the weekend. I see the ocean from my window. There is a lot of nature around, and this really fulfill my wish to always be close to nature and to continually discover nature, both during my
Nice.
personal life and in the laboratory. So I really like to live here.
Nice. So, but what do you miss the most about Italy?
I miss family and friends the most. I was very lucky to have a youth full of friends, and I really maintain many of these contacts. We even have a WhatsApp chat with my high school colleague that we started during COVID, and it's very nice to reconnect after a life, to basically with very different experiences. And well, this we can do this without going to Italy, but when I go to Italy, I can see friends and family in person. I also miss from Italy being in places with a lot of history behind. I think there is a temporal dimension to places which I really enjoy when I walk around in an Italian town or an Italian village. And finally, what I miss are the mountains. As I said, I grew up in the north of Italy. The mountains were my backyard, and I enjoyed very much um, going there. I did some climbing also, and this is really where my heart is in many ways, and I missed a lot. And when I go there, I try to go there all the time.
What would you say to a young Italian who's just starting out in your field?
Well, I would say that the being a scientist is a most rewarding and most beautiful and exciting career. Um, there is always something to discover. It's a great excitement to be the first to know something about the world, the world that surrounds us. Uh, it is great to be surrounded by young people, the student, the postdoc. I don't think there is any other profession that, rather than academic profession, that put you in contact so continuously with so many young people. You are never bored. There is always something new to do, to learn. And it's a wonderful profession. It comes, however, with one drawback, and it's not an insignificant one. It's a very intense career, and can be challenging to balance personal life, family, and work. And this is something that should be considered. However, I don't think there would be a better profession, and I would never change the way I lived.
Grazie mille, Pietro, and congratulations again for your Lifetime Achievement Award.
Thank you.